So
Visible
now
now?
we have Dr. Subrati. Yes, yes, sir.
Okay.
Okay, please start.
Okay, good afternoon, everyone. I'm very much thankful to Dr. Sonia Madam from Basu Patna, Dairy Science College. Uh, I'm really thankful, and also I have got this opportunity to deliver my lecture on very important topic on role of bioactive compounds in fermented foods. So first we should know about the what are the bioactive molecules. So bioactive molecules are the enzymes, organic acids and other chemical compounds which generally microbes produce during their growth cycle. And some of the bioactive enzymes produced by microbes, that may be a lipase, protease, pectinase, streptokinase, etc. So all the enzymes produced by the microbes, they have some kind of uh, commercial applications, or in food industry you can use them for the accelerated cheese ripening or breakdown of some complex materials into the simpler forms. So these are the different biomolecules we generally purify from the bacteria through fermentation process. So bioactive molecules released by the lactic acid bacteria when they grow in the foods. You know, the lactic acid bacteria have a very good proteolytic systems, and they produce different proteolytic enzymes. So that enzymes generally release the proteins. Basically, they break down the proteins into the smaller fragments, and these are called the peptides. And that peptides have a different functions or biofunctional properties. They can control the nutritions by increasing the mineral absorptions, or also they can reduce the oxidative stress by scavenge those free radicals. Even they can control the blood glucose level, or can reduce the cholesterols. So there are different mechanisms different bioactive compounds play to control these kind of different metabolic syndromes or metabolic diseases. Also, bacterial metabolites can improve the cardiovascular functions, like they have anti-thrombotic or anti-hypertensive effect. Uh, then microbial inhibition and immunomodulation means some of the antimicrobial compounds produced by the bacteria, they have antimicrobial effects. And also nowadays, some of the bacteria means the gut, in humans gut, that is full of bacteria in the intestine or smaller part of the intestines, and that gut play a very important role to control your brain. So there is a link between gut and brain. It is called gut brain axis. And some of the peptides, maybe it's opioids like peptides or some other anti-opioids controlling this kind of linkage, or some neurotransmitter produced by the bacteria, they also can make a link through gut and brain. So everyone say that if your gut is healthy, then you are always healthy. Then lactic acid bacteria have been considered for the production of functional biomolecules and food ingredients such as biothickeners, bacteriocins, vitamins, peptides, enzymes, flavoring compounds, organic acids, and amino acids. We will discuss one by one. And these metabolites have found that have a different application in food industry for product manufacturing, or it can improve the product characteristics by improving the textural quality of the product. It can reduce the allergenicity with some of the proteins. Food proteins are there which can cause some allergens to the human being. So some of the enzymes are there which can degrade or hydrolyze those proteins so that they may not have the allergen. Value addition, because you know that lactic acid bacteria, good bacteria, they are called probiotics also if they pass through the ICMR DBT guidelines. And we can add into the fermented foods or fermented milk products to make it value additions. And some of the lactic acid bacteria, they produce the vitamins or some other short chain fatty acids. So it's a value addition to the products. And the preservation, because lactic acid bacteria, it produce the organic acids. It's maybe a lactic acids or acetic acids. Okay, so these organic acids has a very good role to control the pathogens, mainly the foodborne pathogens, and we can preserve the foods for extending the shelf life of the product. Lactic acid bacteria, basically Lactococcus, Streptococcus, Pediococcus, and Lactobacillus. They are commonly used for the basically for milk fermentation. They're very popular in dairy industry. And during the lactic fermentations, they produce variety of metabolites such as organic acids, diacetyl, hydrogen peroxide, bacteriocins, which provide fermented foods distinctive flavors, textures, aromas, while preventing spoilage, extending the shelf life, and inhibiting the pathogenic organisms. Basically, I'm giving an overview about the biomolecules produced by the lactic acid bacteria and which are now being used as a food ingredients for different purposes. If we take the example of lactate, acetate, and propionate, they may be used in food industry to control the acids, or they may be as a pH regulator, also some flavor enhancers. Okay, because in lactic, whenever you have a dahi, you have a clean lactic flavor. Then hydrogen peroxides, it has antimicrobial effects. Then carbon dioxide, it also has antimicrobial effects. It creates the anaerobic conditions. So those bacteria which doesn't like the anaerobic condition, they will get inhibited. And it also contribute to the product characteristics in case of kefir, kumis, Swiss cheese. And then biobacteriocins, this is the another antimicrobial substance produced by the lactic acid bacteria. They are being used for biopreservations, or sometimes it is also being used for the packaging industry to control the growth of the foodborne pathogens. Then diacetyl, it is a very important compound for the fermented milk industry because it gives a buttery aroma of the dairy products such as butter, cultured buttermilk, cottage cheese, and it also has some antimicrobial effects. Then acetaldehyde is the another important flavor for yogurt because diacetyl generally is the main component of the dahi, but acetaldehyde is very important for yogurt preparation and some cheeses. Then beta-galactosidase, it is an enzyme. It is required to break down the lactose. Those have a lactose intolerant problems, they can get the milk free of lactose, or they can have the lactose hydrolyzed milk. So to prepare those lactose hydrolyzed milk, you require these beta-galactosidase enzymes, and lactic acid bacteria produce the beta-galactosidase enzymes. Then proteinase, proteases, peptidases, and lipases, these are the different enzymes produced by the lactic acid bacteria. And these enzymes are basically used to accelerate the cheese ripening, because some of the cheeses which require more ripening period, like 1 year, 2 years, or more than 6 months. But if we use this kind of, because in ripening basically happens the breakdown of the different biomolecules like protein, fat, and carbohydrates. So basically the protein, because in case of cheese, you know that it is a rich source of fat and proteins. So these enzymes can be used to break down those fat and proteins, and you can reduce the ripening time, and you will get the good flavor as well as the very good product. So those products can be easily absorbed in your systems. Then another important biomolecule produced by the lactic acid bacteria is the EPS, exopolysaccharides. You know that exopolysaccharides play a significant role for the improvement of the textural quality of any kind of fermented foods and fermented milks. So basically, they play a role in emulsification, stabilizations, and gelling agents. Also, they can be used as a thickeners and encapsulants. And another important part of biomolecule produced by the lactic acid bacteria is the peptides, or we can say it is a therapeutic peptides, because if we take the example of the milk proteins or any kind of food proteins, the lactic acid bacteria or other yeast or molds which are producing these kind of proteolytic enzymes, they can break down the proteins, and the proteins will get fragmented in a very smaller amino acid residues, and these peptides have a different health benefits like antimicrobial, hypertensive, antioxidative, okay, even anti-diabetic. So these are the different peptides have a different roles, or they have a different beneficial effects on human beings, and these products are also available in the market. So first, which is very important for biomolecule produced by the lactic acid bacteria, is the organic acids. When milk is getting fermented to get the dahi or yogurt, the lactic acid bacteria produce different kinds of organic acids like lactic acid, acetic acid, and formic acid, as well as the propionic acids. And these organic acids bring reduction in the pH of the milk. It influences the subsequent microbial activity in the fermented milk. And at a low pH, a large amount of lactic acid is in the undissociated form, and it is toxic to many bacteria, fungi, and yeasts. Acetic acid and propionic acid produced by the lactic acid bacteria strains may interact with the cell membranes and cause the intracellular acidification and protein denaturation. So basically, you can understand that how that organic acids react with any kind of uh, pathogenic bacteria. So if you want to kill the other organisms with the help of these organic acids, because most of the organic acids they present in the undissociated form. So when they are in the undissociated form, they can easily penetrate inside the cell membrane of the bacteria. And when they goes into the cell or inside the cell, the pH of the inside of the bacteria is around neutral. So these organic acids will get dissociates inside the bacterial cell, and they are being converted into the anion and cations. And then these anionic forms of these organic acids, they basically kill these uh, uh, or disrupt the biochemical processes happening inside the cell. Then acetic acid and propionic acid, they're more effective against some of the pathogenic bacteria because their pK values is very high. It is 3.08 for lactic acid and 4.75 for acetic acid and 4.87 for propionic acids. Acetic acid was more inhibitory than lactic acids because they have a more pK values, and they can also be used to retard the growth or the germination of the Bacillus cereus. You know that it is a very heat resistant organisms. They cannot be killed through the pasteurization. So Bacillus cereus basically it is called spoilage causing organisms. They cause sweet curdling like spoilage to the milk. So we can control this kind of spoilage causing organism through these organic acids. And organic acid also contribute to the sensory attributes of the fermented foods. Again, we have to be very much careful about the sensory quality of the product, because your sensory quality or sensory or you can say the typical taste of the product should not get changed as you are adding some other ingredients. Then hydrogen peroxide, there is another important biomolecule. It has an antimicrobial effect because that they basically result from the oxidation of the sulfhydryl groups, causing denaturation of a number of enzymes, and from the peroxidation of the membrane lipids, increase the membrane permeability. So basically, this hydrogen peroxide, they target the lipid molecule of the membrane. You know, the membrane is made up of two layer bilipid layer of the membrane. So they basically oxidize those lipid molecules, and they disturb the structure of the membrane, and the bacteria will get die. H2 may also act as a precursor for the production of bactericidal free radicals such as superoxide or hydroxyl radicals, which can damage the DNA. So in case of milk, we generally know there is a one very important and popular system called LP system, lactoperoxidase systems. That systems is very much useful for rural areas where the electricity has a big problem to chill the milk during the collection time. So we can use this hydrogen peroxide, and another is the catalase, and there is a thiocyanate. So these are being added into the milk, and these H2O2, they create some free radicals, okay, and that free radicals will damage the DNA, and that bacterial numbers will not get up, so that you can preserve the milk for at least 4 5 hours. So where there is a problem, mainly it is happening in hilly areas where there is a big problem of bulk chilling center to have a very good electricity system. So we can use these kind of LP systems to control, or they have some bactericidal or bacteriostatic effect. It has been reported that the production of hydrogen peroxide by lactobacillus and lactococcus strains, they can inhibit the Staphylococcus aureus, Pseudomonas species, and different cyclotropic microorganisms in foods, because you know the cyclotrops grow at low temperature around 7 to 10 degrees Celsius. So these kind of LP systems are very effective to control those kind of pathogens. Then another important biomolecule is carbon dioxide. Carbon dioxide is mainly produced by the heterofermentative lactic acid bacteria, because lactic acid bacteria are of two types, homo and hetero. You know, the homo fermentative, they don't produce carbon dioxide, they only produce lactic acid, but in heterofermentations, they produce gas, they produce acids, and sometimes they also produce some kind of alcohol also. So it's carbon dioxide, they have an antimicrobial effect because they can create an anaerobic environment which inhibits the enzymatic decarboxylations and accumulation of carbon dioxide in the membrane lipid bilayer, which may cause dysfunction in permeability. Many of the food spoilage microorganisms, especially gram-negative psychotropic bacteria, effectively inhibited by the carbon dioxide. So those products which are being uh, dissolved in the carbon dioxide, they produce the carbonic acid inside the product, and that low pH have a great impact on the bacterial growth, and they can control the spoilage causing organisms, even gram negative bacteria. Why it is so effective against gram negative? Because the gram negative has a good amount of lipid, so this carbon dioxide has a direct effect on the lipid in the cell membrane, so they are more effective against gram negative than the gram positive organisms. The antimicrobial activity of carbon dioxide varies considerably between the organisms, and mostly at 10% level of carbon dioxide could lower the total bacterial counts by 50%, and at 20 to 50% had a strong antifungal effect. So here it's very important that the carbon dioxide can not only kill the bacteria, but the most important part is the fungus. To control the fungus is a very tedious job in any food and dairy industry. So where we whatever the but it's not that you can punch carbon dioxide in any products. Where it is compatible, there you can use it to control the growth of bacteria as well as the fungus. Then flavoring compounds. If we talk about the flavoring compounds, generally in milk fermentations, there are two different fermented flavoring compounds like diacetyl and acetyl, very popular because diacetyl is the prominent flavoring compound of dahi and acetaldehyde is the prominent flavoring compound of the yogurt. So in addition to the catabolism of the milk sugar, basically the lactose, lactic acid bacteria also has the capability to metabolize other substrate like citrate, and citrate is the precursor for production of the diacetyl. Okay, so citrus fermentation by lactic acid bacteria leads to the production of carbon compounds, mainly diacetyl, acetone, and butanediol, which have a very good aromatic properties. They enhance the sensory property of the product. And diacetyl is responsible for the buttery aroma of dairy products such as butter, cultured buttermilk, and even cottage, and most importantly, dahi. Diacetyl is an important component of the flavor of different kind of cheese also. Strains of Lactobacillus lactis, subspecies lactis, and Bioactive diacetyl lactis. It is the strain which produce most of the diacetyl flavor. Apart from these bacteria, another bacteria is there, Leuconostox and Weisselia. Bacilla is a genera which also produce diacetyl during the fermentation of milk. And diacetyl also possesses antimicrobial effect. It is found to be more inhibitory towards gram-negative bacteria than the gram-positive bacteria. And then acetaldehyde. Another one is the acetaldehyde, and it also has a good, very good aroma compound, or the aroma sensing compound, and it is produced specially in yogurt. So lactic acid bacteria can produce acetaldehyde through several pathways, including from the glucose through the pyruvate and acetyl-CoA intermediate of glycolysis, from amino acids and other metabolites that are converted to the pyruvate, or through the conversion of the threonine. So there are different pathways that through which you can produce the acetaldehyde. Okay, so we have to follow the different biochemical reactions. And acetaldehyde at a concentration of 10 to 100 ppm is inhibitory against Staphylococcus aureus, Salmonella typhimurium, and Escherichia coli in dairy products. So it also has a very good impact on against these foodborne pathogens. Then enzymes. So another biomolecules are the enzymes. It is produced by the lactic acid bacteria. So these lactic acid bacteria influence the processing, composition, and organoleptic properties, overall quality, and shelf life of the fermented foods. Lactic acid bacteria can be used as a source for the preparation of enzyme extracts that are able to function under the environmental conditions of the fermentation, and they also release various enzymes into the GI tract and exert the potential synergistic effect on the digestion and alleviate the symptoms of the intestinal malabsorptions. So the enzymes has a very good role in our digestions. All of we know when the food reaches to the stomach, there is some proteolytic enzymes which can work at a low pH like pepsin and trypsin. As it goes to the smaller intestine, you know that smaller intestine is full of bacteria. But those good bacteria like lactic acid bacteria, they produce different kinds of proteolytic enzymes, or they can produce some lactase, so that you can easily hydrolyze those food components. Maybe it's a protein or fat or carbohydrates like lactose, and easily you can digest it. So enzymes has a very good role in our digestion systems. Among the enzymes produced by the lab that most commonly used for the dairy applications include galactosidase, because that will reduce the low lactose products. Then proteinase, it is used for acceleration of cheese ripening. Then proteases for reducing the allergic properties of the cow milk products, because in cow milk there is a one protein called beta lactoglobulin which may cause some kind of allergenicity to the peoples. So we can use this kind of enzymes, proteolytic enzymes, which can break down the beta lactoglobulin, and it doesn't have any allergenicity to the host, human host. Then lipase for the development of the lipolytic flavors in different types of cheeses. So when we are talk about the uh, enzymes, so in our lab we have worked on production of the beta galactosidase enzymes. One of my MTech students, she worked on it. She basically tried to extract the beta galactosidase from Lactobacillus helveticus and other, she took another 6-7 lactic acid bacteria, and she has screened out that which bacteria is giving the best one. So for this, she followed some protocols to extract this beta galactosidase. So first she grows all the cells, means all the lactic acid bacteria well, then collected those cells pellets by through centrifugations, and then cell pellets are washed with a 0.05 molar phosphate buffer, and then she collected all the cells, means the cell pellets, in a buffer, and then she has given the treatment on sonications, because ultrasonication, it ruptured the cell walls, and as you rupture the cell wall, because beta galactosidase, this is a very you can say that it is a limitations for lactic acid bacteria, that the beta galactosidase enzyme cannot come outside the cell because it is an intracellular enzyme. So if you want to extract the beta galactosidase, you have to rupture the cell wall, then only these enzymes will come out from the cell. That's why in industry or in commercial aspects, basically those who are selling the beta galactosidase, if I talk about the Amul is selling the low lactose milk or lactose free milk. They are adding the beta galactosidase enzymes, but that enzymes are being purchased from the Christian Hansen's. And when I asked them that from where you have collected those beta galactosidase, they said that our source is the Cluberomyces, means the yeast, because the yeast basically produce the beta galactosidase outside the cell. So your downstream processings will be a little bit easier to separate the enzyme and the cells. So, but in case of lactic acid bacteria, it is quite difficult, because your all the metabolites, intercellular metabolites, will come out with the enzyme, and then you have to separate out. So your downstream processing will be a bit different compared to the yeast cells. So basically the industry, they when they produce any kind of enzymes or purify the enzymes from the bacterial cells, they generally prefer the extracellular enzymes. They don't prefer the intercellular. But here we have started, because we are thinking that how can we produce beta galactosidase from the lactobacillus. So we have used the sonications, and after sonication we got the crude extract of the partially purified beta galactosidase, and we have added into the milk, and for 3 different cultures, and we have found that the lactose reduction means the percentage of lactose present in the milk. So in V3, if we adding the 2% partially purified beta galactosidase, the lactose reductions or lactose breakdown is quite high, around 40%. And as the lactose is breaking down, then obviously glucose and galactose will be produced. So you can see here again the culture V3 is the Lactobacillus helveticus. So these cultures again, they produce these around 8% glucose and around 12% galactose. Okay, so we have estimated through HPLC, and these lactose are being broken down into glucose and galactose. So we have tried basically we have tried to see that really it is happening, that after adding these partially purified beta galactose in the milk, first we have sterilized the milk, then we have added these enzymes and incubated at around 30 degrees Celsius, and then we have analyzed after 6 hours or 12 hours and 8 hours, and we have seen that the breakdown of lactose is happening, as well as the glucose and galactose production is also increasing slowly. Then therapeutic peptides. Basically, tomorrow I will take one more lecture on beta, the therapeutic peptides or the bioactive peptides. So I will not tell you in detail about these peptides, but you know that peptides are basically small fragments of the proteins which are being released or secreted by the lactic acid bacteria. They produce different proteolytic enzymes, and all these smaller fragment of the peptides has a good impact on human health. And these peptides are called bioactive peptides, and they can be generated by the digestive enzymes and even in the milk fermentation or with the starter culture traditionally employed by the dairy industry. So bioactive peptides identify. Any questions? So bioactive peptides identified in fermented milk products. Okay. Basically, you can see that these are the different fermented products like cheese. There are varieties of cheese, then fermented milks like yogurt, sour milk, dahi. And you can see these are the different fragments generally generated from the milk protein caseins, mainly caseins. And these peptides fragments are different biofunctional properties like anti-hypertase inhibitor, means angioconverting enzyme inhibitions, or anti-hypertensive or immunostimulatory, okay, or antimicrobial. So these peptides are having a different roles to play when people consume these kind of products. Then bacteriocins. Bacteriocins is a another antimicrobial substance produced by the lactic acid bacteria, and they can be suitable option and are finding application as a biopreservative to enhance the safety and extend the shelf life of products. So when you want to increase the shelf life of products, the bacteriocin is a good options for you to add into the foods, and it can enhance the shelf life because bacteriocins mostly can kill the gram positive as well as the gram negative depending on the source. If the bacteriocin is produced by the gram positive, they can kill only gram positive, not gram negative. But if the source is gram negative, like E. coli, if they produce coalitions, so it's a bacteriocin produced by the gram negative, so it can only kill the gram negative. So this is the limitation of the bacteriocins. But the positive point of the bacteriocin is that these are food grade. And second important part is that that doesn't have any color and flavor. So you can add in any foods wherever it is possible, and they can control the different foodborne pathogens, and it increases the shelf life of the foods. Among the bacteriocins of lactic acid bacteria, nisin is the single bacteriocin which has found commercial application as a biopreservative. So nisin is the first approved bacteriocin by the FDA and WHO, and it has a GRAS status, generally recognized as safe. And the producer organism is Lactobacillus lactis, subspecies lactis, and it is available in the market in different names, trade names. Okay, so you can purchase it, you can add into the paneer or other milk products where it is possible to apply on the basis of the compatibility of the product, and then you can enhance the shelf life of the product. It is used in the pasteurized processed cheese products to prevent the outgrowth of spores, mainly the Clostridium tyrobuticum, because that spore is very much heat resistant. They can survive in at 85 to 105 degrees Celsius. And this Clostridium tyrobuticum, this is a anaerobic spore formers. So these bacteriocins has a good impact on the Clostridium, even in the bacillus also. So bacteriocin is a good option for controlling these spores. How the bacteriocin works? Bacteriocin basically, you can see here this green cylindrical form. Basically, it is a pictorial diagram, not exactly the happen in the cell, but to make it understanding how the bacteriocin works. So basically, this is a green cylindrical portion of the bacteriocins. It gets attached with the cell wall first through docking, and then these lipid molecules are basically they disturb the lipid bilayer, and they make a channel through the cell membrane. And as the channel forms, because there is a channel, or you can say it is a pore formations, and all the material present inside the cell, they will come out and bacteria will get die. So this is the basic mechanism that how these bacteriocins get basically they works and they kill the bacteria. Then another is the vitamins. Certain lactic acid bacteria, not all the bacteria produce the vitamin, but generally certain bacteria produce the B vitamins. This is a water soluble vitamins. And the lactic acid bacteria used in the dairy industry, basically Lactobacillus lactis, Lactobacillus acidophilus, Lactobacillus Delbrueckii subspecies bulgaricus, and Streptococcus thermophilus. So these are the very popular and very important lactic acid bacteria generally used for milk industry or dairy industry. And they have been shown to synthesize folate, and they secrete into the medium during the fermentations and improving the folate content of the products. So vitamin B12, an important cofactor for the metabolism of the fatty acids, amino acids, carbohydrates, and even nucleic acids. So bacteria also require, basically the propionibacteria, they require B12 for different biochemical purposes because it acts as a cofactors. Even in the electron transport chain, if you see the electron transport chain through which bacteria produce the ATP, the electron carriers are basically FMN, flavin mononucleotide. So these kind of vitamins are there. So bacteria requires those vitamins for their own biochemical purposes also. Then genera like Lactococcus, Lactobacillus, Leuconostoc, Bifidobacterium, Enterococcus, they also synthesize vitamin K. And vitamin producing strains may be useful as dairy starters to increase the vitamin content in the fermented dairy products. Vitamin production by lactic acid bacteria is considered a strain dependent trait, and the selection of an adequate combination of strains is essential to develop the fermented foods with increased vitamin concentrations. Besides riboflavin, folate, and vitamin B12, increased levels of other vitamin group vitamins such as niacin, pyridoxine have also been reported as a result of the lactic acid bacteria fermentation in yogurt, cheese, and other fermented foods. Then another important biomolecule is the fatty acids. Under certain conditions, some lactobacilli and lactococci possessing the lipolytic activities, they produce the enzyme lipase in significant amounts, and then they release the fatty acids from the fat present in the milk during the fermentation. But the unsaturated fatty acids are active against the gram-positive bacteria, and the antifungal activity of the fatty acid is dependent on the chain length, concentration, and pH of the medium. The antimicrobial action of these fatty acids have been thought to be due to the undissociated molecule, not the anion, since the pH had profound effects on their activity with a more rapid killing effect at a low pH. We have also worked to produce the vitamins as well as the short chain fatty acids in our lab with a project it's a DBT project funded by DBT with Northeastern Hill University, Shillong, and we have isolated some lactic acid bacteria from their fermented foods, which are basically present or popular in Meghalaya, mainly the Khasi tribes. So we have isolated 5 potent lactobacillus cultures: KGL2, KGL3, KGL4, RNS4, and WTS4. So basically, KGL2 is the Lactobacillus fermentum, KGL3 is the Lactobacillus plantarum, and KGL4 Lactobacillus fermentum, RNS4 is the Lactobacillus rhamnosus, and WTS4 is the Lactobacillus fermentum. So these 5 lactobacillus cultures were considered for the vitamin production, B2, B12, and B9. And also we have checked the short chain fatty acids. Okay, so the KGL3 had shown the highest vitamin B2 productions, but KGL2 exhibited maximum B12 productions after 36 hours when we are growing in a vitamin-free media, basically basal media. And WTS4 produced the maximum folate around 0.09 microgram per ml after 24 hours. And RNS4 produced maximum short chain fatty acid compared to the other 4 lactic acid bacteria, mainly acetic acid, lactic acid, and butyrate. So we have worked on the lactic acid bacteria for production of the short chain fatty acids and the vitamins. Okay, but it is in vitro conditions, not in a product format. Okay, so here we have taken the cultures individually. We have grown in a vitamin-free media. It is available through high media, and we have checked the vitamin production through HPLC method by comparing with the standard. Then another important biomolecule is the CLA, conjugated linoleic acids. Lactic acid bacteria also produce the CLA, like Lactobacillus and Streptococcus. Basically, the biosynthesis CLA, which may have a positive effects on inflammations, cancer, metabolic disorders like insulin resistance, body weight control, and cardiovascular diseases. Some isomers of the CLA reduce the carcinogenesis and body fat. Also, CLA concentration play a role in the balance toward beneficial or detrimental effect. Anti-carcinogenic effects are observed at CLA doses of 0.5 to 1% of the total diet, and studies showed that the humans generally excrete 20 milligram of linoleic acid per day. So CL is very much important for our body to prevent different kinds of metabolic syndromes. Then bacteria or lactic acid bacteria also produce the sweeteners, okay, like L-alanine productions. So conversion of pyruvate into the alanine takes place in various anaerobic bacteria, and it involves a single enzymatic step catalyzed by the alanine dehydrogenase. So that enzyme is very much required to produce the alanine from the pyruvate. The pathway was introduced in Lactobacillus lactis through cloning and expression of the ALAD gene of the Bacillus sphaericus that encodes for the L-alanine dehydrogenase with a kinetic property similar to those of the LDH of Lactobacillus lactis. So basically, through cloning, that gene ALD, which is responsible for alanine dehydrogenase being transferred to the Lactobacillus lactis, and that Lactobacillus lactis produced the L-alanine. ALAD was cloned under the control of NICE promoter in Lactobacillus lactis, and its overexpression resulted in a 30 to 40% rerouting effectively from lactate toward alanine when resting cells grown on glucose were used in the presence of excess of the ammonium, which is required for the conversion of pyruvate to alanine by ALAD gene. And furthermore, introduction of the system into LDH deficient lactobacillus cells resulted in the complete conversion of glucose into the alanine by changing the metabolism of Lactobacillus lactis from homolactic to the homoalanine. So basically, it is a gene cloning systems through which you can take the alanine D gene from the Bacillus sphaericus, and you can transfer those genes into the Lactobacillus lactis through NICE promoter or through different vectors, and then these genes can be replicated inside the Lactobacillus lactis, and you can increase the yield of the L-alanine productions. Another sweetener is the mannitol. There is a study conducted by Arini Kanas in 2007. So they constructed a strain of Lactobacillus fermentum through inactivation first of IDH, LDHD, and then of the LDH L gene that was able to produce mannitol and either L-lactate or mannitol and pyruvate in a single process. The process was anaerobic, and the recombinant produced the 2,3-butanediol as a co-product. But Helena et al. 2003, it reported that yield of mannitol from fructose from Leuconostoc pseudomesenteroids was improved from 74 to 85% by applying random mutagenesis to the producer organism for inactivating its fructokinase activity. So through genetical manipulations, you can increase the yield of these mannitol. Then sorbitol. Nissan et al. In 2005, they constructed lactobacillus acesi strain in which the sorbitol 6p dehydrogenase gene GATF was integrated into the chromosomal lactate through lac operant systems, and the gene was under the control of the lac operant, which is repressed by the glucose and induced by the lactose. The recombinant cells pre-grown on the lactose and produced 0.024 molecule sorbitol per molecule of the glucose, while the parental strain produced very negligible amount of the sorbitol. So by producing or by transferring those or by overexpressing those genes, basically the sorbitol 6PD hydrogenase genes through the lac operant systems, the lac operant basically control the lactose metabolisms. So on the basis of the lactose metabolism, you can increase these sorbitol content. Then mannitol phosphate dehydrogenase and LDH were inactivated in a lactobacillus plantarum strain. Overexpression is sorbitol dehydrogenase gene, as both MDPH and SDH use fructose 6-phosphate as a substrate, and the inactivation of the MPDase allowed more F6P. F6P means the fructose 6-phosphate to be reduced to sorbitol 6-phosphate by the SDH, sorbitol dehydrogenase. The result in resting cells with the glucose as a substrate, and there was a near to theoretical yield of sorbitol around 0.65 molar per mole of glucose. So by only cloning systems, you can increase the particular sweetener. Then another important biomolecule, that is exopolysaccharides, which is also produced by the lactic acid bacteria when they are growing during the fermentation of milk, or we can use the culture as a EPS producer also through fermentation systems. So exopolysaccharides are formed monosaccharide residues of sugar and sugar derivatives. They are produced by plants, algae, fungi, and bacteria. During the growth, EPS is secreted into the external environment by microbes, and distinguishing EPS from capsular polysaccharides, which are cell bound. So basically, very important aspects of the EPS that they are going outside the cell. So it is basically extracellular material. The EPS produced by lactic acid bacteria or EPS producing strains of lactic acid bacteria can be used directly in fermented dairy food productions because of the generally regarded as safe, because they have a GRAS status. They can be directly used in the food. Streptococcus, Lactococcus, Pediococcus, Lactobacillus, Leuconostoc, Vacella species among lactic acid bacteria strains are frequently produce the exopolysaccharides. Also, some non-starter lactic acid bacteria like bifidobacteria are able to produce the exopolysaccharides. So, exopolysaccharides from lactic acid bacteria prove to have a immunomodulatory, anti-tumor, cholesterol lowering, antioxidant, prebiotic, and antibacterial effects, and have been found to increase the colonization of probiotic bacteria in the gastrointestinal tract. So, EPS has a very good health effects. It can have a colonization effect in the gut. The bacteria can produce this kind of uh, materials, oligosaccharides materials. And in consideration of the characteristics of exopolysaccharides, they are largely varied in molecular structure, mass molecular size, charge, and as a consequence in their rheological properties. Exopolysaccharides from lactic acid bacteria are highly various polymers and can be classified following different criteria. One of the most used classification criteria based on their monomer compositions, means what are the material present in the exopolysaccharide. So basically, depending on the composition of the repeating units and biosynthesis pathways, EPS can be classified by two parts. One is called homopolysaccharide, another is called heteropolysaccharide. So homopolysaccharide, you can see here from this chain that one monomer is repeating several times. So it is a homopolysaccharide. So it is a linear chain, but here also the same monomer, but they are repeating in a linear chain as well as from the branch chain. So it is a branch chain homopolysaccharides. But one monomer means maybe it's a glucose or it's a sucrose. They're repeating in a sequence. Okay, so they are homopolysaccharides. So what are the examples of the homopolysaccharides produced by the lactic acid bacteria? Like alpha deglucans, they basically dextran, mutan, alternan, reutran, and they are produced by Leuconostoc mesenteroids, Leuconostoc mesenteroids subspecies dextranicum, then mutant Streptococcus mutans or Streptococcus sabrinas, then alternate Leuconostoc mesenteroids, reutran Lactobacillus reuteri, then beta glucan, it is produced by Pediococcus or Streptococcus, then under fructans, levans, and inulin type homopolysaccharides are there. Streptococcus salivarius, Streptococcus mutans, Leuconostoc citrinum, Lactobacillus reuteri, then polygalactans. This is produced by the Lactococcus lactis subspecies lactis or Lactobacillus delbruecae subspecies bulgaricus. Then heteropolysaccharides. Heteropolysaccharides, you can see that there is a linear chain heteropolysaccharides or branch chain heteropolysaccharides, but you can see the monomers are changing. There are two different monomers are repeating in this line or in this chain. So the majority of exopolysaccharides produced by lactic acid bacteria are heteropolysaccharides. They containing 3 to 8 repeating units composed of 2 or more monosaccharides such as glucose or galactose, rhamnose or mannose, and sugar derivatives such as glucouronic acids, N-acetyl glucosamine, N-acetyl galactosamine. And lactic acid bacteria which produce the heteropolysaccharides are Lactobacillus rhamnosus, Lactococcus lactis subspecies lactis, Lactobacillus casei, then Streptococcus thermophilus, Lactobacillus helveticus, Lactobacillus acidophilus, Lactobacillus delbruecae subspecies bulgaricus. So there comes under thermophilic lactic acid bacteria now on the previous one for mesophilic. So they all produce the heteropolysaccharides EPS, and their molecular mass of the heteropolysaccharide range from 10 to the power 4 to 10 to the power 6 Daltons. Basically, Gillians, xanthan, and kefiran are some of the example of heteropolysaccharides. So how these exopolysaccharides produce, isolate, and quantified and characterized? First, this is the cultivation of EPS producing strains. So first you have to find out the EPS producing strains by deciding some assays which will give you the best judgment of EPS producing strains. So then after screening, you can select them and then you can optimize their productions. Means or you can optimize their growth conditions for maximum production of the EPS through food matrices or in a medium. Okay, then you have to, after productions, you have to go for isolation and recovery of the exopolysaccharide. So there are different methods are there by through you can, because from where the downstream processing will start, and you can purify those EPS from those complex materials. After getting the purified form of the EPS, you can quantify them, you can characterize them by using different kinds of techniques like gravimetric, colorimetrics, or FTIR, NMR. Okay, so there are different methods are there for quantification and characterization. So how you produce the EPS? For the EPS production, 5% skim meal, 0.35% yeast extract, 0.35% peptone, and 5% sucrose was inoculated with an overnight culture and incubated for 29 hours at 30 degrees Celsius. Bacterial cells were removed by centrifugation at 4 degrees Celsius after centrifuging at 9,000 RPM for 20 minutes. And then TCA, trichloric acetic acid, was added to the supernatant at a final concentration of 4%. And then precipitated proteins were removed by centrifugations. And the clear supernatant was collected and precipitated by adding the double volume of cold absolute ethanol and maintaining the overnight at 4 degrees Celsius. So precipitate was recovered by centrifugation at 9,000 RPM for 20 minutes at 4 degrees Celsius. And then the precipitate was dissolved in the ultra pure water and dialyzed for 2 days at 4 degrees Celsius against the same solutions, using dialysis membrane having a cutoff of 12 to 14 kilodalton. After dialysis, the solution was frozen at minus 80 degrees Celsius and lyophilized at minus 50 degrees Celsius for 3 days to completely remove the solvent. So this is the process to produce the EPS, and this is the downstream process to recover the EPS through centrifugation or filtrations or recovery of the polymer from the cell-free supernatant, then precipitation as we discussed, and then precipitate can be separated from the solvent through the drying systems. So this is the downstream processing through which you can purify the EPS, exopolysaccharides, and maybe it's a homopolysaccharide or heteropolysaccharides, and then you can store it for different storage conditions. So finally, I am concluding my sessions here. The functional biomolecules and food ingredients of lactic acid bacteria are increasingly finding application in the food industry for reasons such as increased demand for the health foods, novel foods, and natural products. Also, the use of antimicrobial biomolecules as a natural preservative in food, as a novel way of food preservation, and it has much gained attention in recent years for producing the fresh products or novel products by adding those biomolecules, or also to extend the health benefits of the functional biomolecules by adding the probiotics or their different biometabolites or biomolecules. Okay, so this is the conclusion of the topic, that these kind of biomolecules can be used as a food ingredients for the production of the food, so that we can use it for any kind of food formulation or any kind of functional food development. So thank you all for listening my lectures carefully, and now
Thank you, sir.
open for discussion.
Participants, if you have any questions, please ask.
Thank you. And I invite all of them for question session. So if you have any confusion regarding that presentation, you may ask in the inbox or